Okay, so I'm just going to talk to you a bit about proteogenomics. I don't know if this is working. Okay, so just some basics on proteogenomics. It's the intersection of proteomics and genomics, as the name alludes to. So as genomics has become a lot cheaper, sequencing is much easier to do for all sorts of different experiments, right? Which I'm sure you guys have heard about extensively in some of your other classes. So in combination with mass spec, um, the sequencing can actually be used to help genome annotation to study the effect of genomic variation within the proteome and also in biomarker identification. So together they're a really powerful pair. And proteogenomics, the word proteogenomics was first published in 2004 in a paper about proteogenomic mapping to help inform genomic annotation. And since then, it's been used in a variety of different studies. This is an older review, so it doesn't actually include everything, but it's become a much more popular method. So let's just go through the basic workflow. As with all of our proteomics, there's high throughput MSMS, which you've heard all about. You get your spectra. And as you've probably heard about before with David, you can either do de novo sequencing, which is actually what Trixie is going to talk to you about in a lot of detail. But de novo sequencing, you don't require any knowledge about the peptides that might be present in the sample, and you just use mass differences to determine like each amino acid that occurs in the peptide chain. This is it can be a pretty slow process, but people like Trixie are experts at this. And you can also do protein sequence database searches where you have a sequence database, so it's a FASTA protein or peptide database, and you essentially take your spectra and you compare it to your database, and then you find matches based on that comparison. And then you can also do a spectral library search, which was similar to what Jessica was just talking about, where you have your query spectra and then you have a library of spectra that other people have gotten experimentally, and you can compare those two and find matching peptides based on that. So those are the three sort of ways of finding peptide identification. Here we're going to focus primarily on this protein sequence database search. And this is where proteogenomics really comes into play. So there are three requirements for proteogenomic analysis. You need to have DNA or RNA sequencing data. You need high-resolution MSMS. And you need informatics tools for both proteogenomic database construction and also protein searching. So if you have a sample, you do DNA and or RNA sequencing on that sample, and you also do MSMS on that sample. So using the sequencing data and informatics tools that we'll discuss, you can create a protein database based on the DNA and RNA. So not the reference database, but something that actually takes the DNA and RNA information and determines hypothetical proteins that are going to be in that sample. And then you have your mass spectra, and you compare these two to identify peptides and proteins within that sample. And this also uses informatics tools such as Xtandem. I think you've probably heard a lot about these. These are the same tools that we would use here. You would just use a protein database that's different and not the reference. And that's really like the primary thing here. We're going to go into a lot of detail about that. So in the past, computational algorithms were commonly used to predict and annotate genes, but there were quite a few limitations surrounding these algorithms. And with mass spec, we can both confirm existing gene models and also correct the gene models that we have, and then also identify some novel genes and splice isoforms based on the proteins that we find that are actually within our sample. So this is just an outline of what we'll be going over. How proteogenomics can be used to improve genome annotation, to also create these sequencing-driven databases, the construction of these, and then how we can map our proteins back onto genomic coordinates to sort of look at all of our data at the same time. So just to start with genome annotation, what the definition of genome annotation is: the process of identifying and assigning function to genes. Historically, this has been done either using some sort of comparative sequence similarity. So if you have, a similar species, saying that we likely will have similar exons and proteins within this other species. Gene prediction algorithms that are done computationally, and RNA transcript analysis. And there are, of course, limitations with all of these methods. A lot of them have issues finding gene start and stop sites, the reading frames, identifying short genes and overlapping genes, and then, of course, alternative splice boundaries. And then, with RNA transcript analysis, just because it's transcribed doesn't mean it's translated. So that's another big issue when it comes to using these for genome annotation. So MS-based proteomics can really be used to supplement the sequence analysis for genome annotation. So just a little bit of background on protein sequence databases. So these protein sequence databases are just FASTA files that have what you assume is in your sample, right? So if you are missing peptides in that database, you're never going to find them in your mass spec, right? So that's really the main thing. So, but then again, if you include everything in your database, then you're searching a huge data set and you're going to lose a lot of sensitivity. So you really want to balance out including as much as you can, but not including like everything every peptide or protein that could possibly exist in the world, right? Because then you're going to have a huge data set to search, and you're just not going to have any sensitivity. So it's sort of that balancing that you're really working with here. Okay, so in a normal mass spec experiment, you have your spectra, and you use some sort of reference database, RefSeq or Ensemble or something, and through this you find your annotated peptides. With proteogenomics, very often we have a 6-frame translation database, which I'll go into detail, where we can then, and these are commonly used to search, these 6-frame translation databases are commonly used if you want to remove all of the bias, because you're essentially translating every possibility from your genomic data, and I'm going to go through an example of that, but you're essentially taking out all of the bias that you have about what you think is already there, which is what's in the reference database. So here you can find both annotated and novel peptides using a 6-frame translation. So what is a 6-frame translation? Okay, so say you have your genomic, this is your genomic sequence from exome sequencing, or whole genome sequencing. You have your positive strand, and then there are 3 options of where to start on the positive strand, right? You can start with ATG, you can start with TGA, or you can start with GAA. These will all give you completely different sequences, so that's 3 of them. And then on your negative strand, you then start, it's essentially the same thing, but going backwards, right? So the 6-frame translation takes all of the genomic data and does this to it. So it's actually a huge database, but if you know nothing about your sample, this is sort of the way to start. So there's 3 different softwares available that do this, where you can sort of input your genomic data and it outputs this and also does the MS searches for you. You can also easily write a Python script that does this. This is a pretty easy thing to do. So just an example of how we can use genomics to better annotate our genomes. So this is using a species of mosquito. This is just an example I got from a review. And I'm sorry, it's a little hard to see, but in this case, this vector-based gene here is the predicted genes based on what they already had annotated. So here they find a peptide that matches this annotated or predicted gene. Great. But they also find peptides that match up to this 3' untranslated region, sort of implying that there is this extra exon before our predicted gene region. Additionally, another example here, we have our gene predictions in orange, and we find peptides matching to gene predictions, and then peptides matching to an unannotated gene. So this is gene predictions using like the computational method. Here's what's in the current reference, this red. So you can see we're getting our current reference peptides out, but we're also getting a whole new, 2 new exonic regions here. And then here we have, there's no actual, there's nothing in the reference here. So this is just, within some sort of intronic space, and we find peptides that match up to some other, unknown, completely novel gene. And this is just showing that there's a conserved open reading frame in a related species. And this is just the sequence of the species of interest and our related species, showing that that makes sense. They've shown that this exists in a related species, and we're finding peptides that match it in our species. So those are some examples. And this is a little more of a broad example of all of the things that you can find using proteogenomics. So here, let's say this is our annotated gene. We have some exons. We have a hypothetical reading frame. We have a splice isoform, right? That's what we have. So this is what we find in our proteomics. This is where all of our peptides match. So first of all, in A, we find some upstream peptide, which is showing that there's actually a new transcriptional start location. It wasn't actually here. In B, we actually are able to confirm this hypothetical open reading frame. In C, we confirm the splice junction that was hypothetical here. In D, we actually identify that this is not actually the right reading frame. It's a different reading frame. In E, we actually find a completely new exon. In F, we confirm that this is actually where the gene ends. And then in G, we can see in a related species, we find this new, this novel exon here, but in another related species, we actually see that that's conserved, so that makes sense. So this is just this is obviously not a real example, but just showing all of the things that proteogenomics can do in terms of genomic annotation. Okay. So in addition to the 6-frame translation database, you can also use RNA-seq so RNA-seq will find junctions, it will find the intron-exon boundaries, and you can throw all of that into your database, which we'll go into detail about how that happens, and find novel splice isoforms, which is a really powerful method. Let's say you don't have any genomic data on your species, you can take a related species genomic data and see what you find from that. So there's an example in the Fennu lab where they're using the mouse database of the mouse protein sequence database and genome database to try and find things in rat, right? Because there's been way more studies in mouse than rat. So things like that. So just to summarize the genomic annotation part of proteogenomics, so we can confirm existing gene models, we can correct for the gene models based on both intron-exon boundaries, reading frames, novel splice isoforms, and novel exons. We can identify completely novel genes. We can identify fusion proteins, which I will discuss in a bit more detail as well. And we can identify genomic polymorphisms such as single SNVs, and et cetera, which we're going to go into detail about as well. So let's talk a little bit about the sequence-driven database construction, which is really the primary thing here, even with genomic annotation. So you saw this before, but before proteogenomics, the main way that we were looking for these variant proteins, so proteins that were not in the reference database, was using this de novo sequencing, which, as I mentioned, is very time-consuming. So very few peptides were sequenced in this way. So instead, if we add in our DNA and RNA sequencing into our protein sequence database step, we're really able to make this a more automated process. Okay, so let's start with looking for single nucleotide variants. So single nucleotide changes within the DNA that result in some sort of downstream protein change. So here, let's say we have all of the genomics SNVs that we found from our sequencing data. We can add those into we have a reference database and we add those in so that what we predict in our protein to occur based on these variants. And we can then identify variant proteins using a combination of these databases. So how do we do that? I don't know if you guys have used a VCF file. This is the output of genomic analysis looking specifically for SNVs and other indels, etc. But let's just focus on the SNVs. So we use this. We essentially extract from this the position, the chromosome and the position, and the new nucleotide that it's changed to, right? So we use that information. And then we create our variant sequence database by taking, okay, let's say this is our reference genome sequence. We add in the variants that we found here. And then we do an in silico translation. And then we create our variant protein sequence database containing all of our variant peptides, essentially. And from here, we can then search our MSMS and find if this peptide exists. We would never have found it before, right? But now we can actually identify it. So similarly, we can use RNA-seq data to find novel splice junctions and alternative splicing. So we, again, can take RNA-seq junction data. So in RNA-seq, you can find the intron exon boundaries, and then we can essentially filter out everything that's been already identified and look for only things that are new, right? So either unannotated alternative splicing, novel expression. I'll show you some more examples of that. But what comes out of an RNA-seq experiment is, well, if you do specific top hat analysis, is a bed file of the junctions. So it's hard to see here, but you essentially get the chromosome number and then the start and end of where the intron exon boundaries are. And we can just use those genomic locations to then see, okay, have these intron exon boundaries been reported before? If not, let's add them into our database. So we map them to known boundaries, filter those out, and then we have a bed file that contains only novel intron exon boundaries. And those can really be described in 3 different ways, either as unannotated alternative splicing. So let's say we know that exon 1 and 3 exist, but we don't know that they connect. So that's one example. We can also have one novel boundary region. So we know exon 1 exists, and we have one end of our boundary here, but then we actually have a shorter exon 3 occurring, or maybe it's in an intronic region. Maybe it's out in the middle of nowhere. Or we have 2 novel intron exon boundaries. So there's really 3 different ways of sort of categorizing these. But we can do that and then essentially do another similar in silico translation of how these would look, as proteins. So the third thing I'll discuss is fusion protein identification. This is primarily in cancer studies when you have fusion genes. So let's say our genomic analysis showed that gene X, exon 1 of gene X, was attached to exon 2 of gene Y. We can just essentially translate what that would look like and then see if we find it. Yeah, go ahead.
Oh,
no, it's fine.
In your previous example, you show a genes, you can only map the second part of the.
Yeah.
That could be the fusion gene.
Yeah, well, fusion genes it's true that fusion genes are specifically when they're like really far away. That's how they categorize them, right? So either they're on different chromosomes or they're so far that like they're overlapping a ton of genes. So there's like a specific way that the computational tools identify fusion genes that don't necessarily get picked up by this. So you sort of have to do both to get like all of the information. If you're doing, let's say, a cancer genomic study, you'd want to do both of those things. Yeah. Okay, so this is what the output of what we were looking at, which is top hat output. So you have this is sequencing, overlapping sequencing data for 1/2 of a fusion gene. So you can kind of see the sequence here, right? And then you have the second part here. So it's just showing sort of evidence of the overlapping genomic sequence. Starting, let's say this was like one part of the exon moving across the exon, and then here from this part of gene 2 across. So you can, from this, find a consensus sequence, and then do a 6-frame translation. Because in this case, you have no idea where anything starts, because it's sort of a wacky example of what happens in biology. And then from here, you would then use this to then search your MS data and see if you actually find any of these fusion proteins in your actual data. So there's some informatics tools to do this. Quilts, which both David and I have worked on, which generates databases from genomic and RNA sequencing data. It outputs FASTA files. There's another program called Custom ProDB, which does similar work with RNA-seq data. And then a Splice graph database creation, which I'm not sure if that's actually open source yet, but I know it is either in the works or has already been, is out in the world. So just an example of how this would be used in the real world biology world. Proteogenomics in human disease. So the 1,000 Genomes project, which I'm sure you've all heard about, did whole genome sequencing on a ton of people and uncovered millions of germline variants that are different between individuals. And we typically use this reference database, RefSeq or Ensemble or UniProt, to model the general population. But if we've shown that in the real world, there's a ton of difference just in germline variants, where we would clearly miss a lot if we were using a reference database, right? We're going to miss even just normal proteins that just have an SNV in them, we're going to miss. So in addition to that, particularly with cancer is, of course, characterized by expression of tumor drivers and suppressors that result from gene mutations, right? So, and these mutations, of course, can influence diagnosis, prognosis, and treatment. An example of that is the BRCA gene in breast cancer. So, cancer proteomics, what we really want to know is, are the genomic variants we find actually at the protein level? If they are, what's their effect on the function of the protein? And can we classify tumors based on these protein markers and use that for treatment? So in addition to just people having a whole lot of variation in their genome, it's also been shown that tumors, even that are within the same tumor type, so here's all breast cancer tumors showing rearrangements, that's what these lines are, just showing that there's a huge element of variation across the same tumor type. So if we really want to study tumors at the proteome level, we need to do a sample-specific protein databases to capture all of this variation. So what we would do is we would have our tumor sample, which we would do high-throughput MSMS, and we would use the germline and the somatic variants within that sample and throw those into our database, right? So that we would not only find the proteins that are annotated at the reference level, but also find tumor-specific and patient-specific peptides based on the germline and somatic variants within that sample. So, so how do we do that? So we have a non-tumor sample. That's where we get our germline. So germline just means, you know, like the normal variants that we have in our body, not the tumor variants. So we use that to find just the variants within your normal cells. Then the tumor sample, which we would use to find alternative splicing within the tumor, somatic variants, and novel expression. And we put all of that into our protein database. Again, this is just some of the other, so we would have variants, we would have alternative splicing and novel expression, and also fusion genes all thrown into here, in addition to our reference database. So together, all of these would be able to identify what we would think is the complete proteome of that tumor. So if you think about it, the tumor-specific peptides that we identify could be used as drug targets, because if it's truly only in the cancer, that would be wonderful. Like if we could find a bunch of proteins that were only expressed in a tumor, that would be like a wonderful drug target. So things like fusion proteins that don't exist normally, novel protein isoforms, or specific variants that are occurring. And also, if we know what genomic variants are actually getting to the protein level, it will really help to figure out what is driving cancer biology, or even like what genomic rearrangements cause proteins to not be expressed at all. Both of those things are really important in this question. So that's the main thing about how we can use sequence-driven database construction to identify novel peptides. Does anyone have any questions about that? Okay. Lastly, I'll just talk a bit about proteomic mapping to genomic coordinates. So once we've identified peptides from our mass spec using genomic data, it would be really great, right, to map them back onto the genomic location. But this requires tools to convert proteomic data to genomic data. And we want to be able to determine the location of the peptide within an exon, whether or not it's proteotypic, if it's a novel coding region. And the main thing is visualizing in genome browsers that already exist, like UCSF and the IGV. And also using this to do some quantitative comparison. So let's say we know that there's a copy number variation at this region in your genome. Does that mean that there's an increase in protein at that region? Do those things happen? Do they coordinate, or are we seeing something completely different? So there's two tools that I know of that do this currently. One is called PGX. It's a Python-based tool that maps peptides back. So it will give you the genomic coordinates of a peptide. This was created by Manor Ashkenazi and David. And then also the Proteogenomic mapping tool, which is a Java-based tool that searches against a 6 reading frame database. So the PGX, you can give them any database you want, whatever you used. And then this one requires a 6 reading frame database. So here's just what PGX looks like. It's on David's website. You put in your peptides and your database, and it gives you genomic coordinates. And this is just looking at an example of what you can do here. You can map out the genomic variation data, the copy number variation, the expression at the RNA level, and then also your peptide expression. You can then zoom in a little bit and just look at how things are changing either together or, apart. And you can also map these onto, this is IGV. So here we have a reference gene. And then here, this is actually an example of a variant peptide. So it has a single amino acid change. This was our reference. This is the variant we found in our genomic data. This is what the peptide looks like after we've added in that variant. And we can map that. We can map our variant peptide here with a reference peptide here and just sort of look at them all in one at once. And this is just where the variant is. And this is our spectra, which we won't go through, but just showing that this actually happened. And then here's an example of a novel peptide. So here we have the predicted exon region in our ensemble gene. We have the red is what the RNA-seq said. Okay, the RNA-seq said, yes, one boundary is here and one boundary is here in this intron. And we identified in our mass spec data a peptide that bridges this splicing region. And then you can also put all of the data together. This is just showing, this is actually like proteomic quantitation data here. This is the RNA-seq expression data, and then you have your reference data and your peptide data from all different experiments. So you can just showing that you can throw a whole bunch of this together if you really want to and see it all in one at once, which I think is really useful in these kinds of experiments. This is actually, so we use PGX to get the genomic coordinates, and then we use IGV to then input all of that data. But you need to first do that first step because otherwise it doesn't like read proteomics data. Yeah. So just to summarize all of this, integrating proteomics and genomics can improve our understanding of not only genomic annotation, but also functional protein products that are integral in biological processes. And proteogenomics is currently being used extensively in cancer discovery. Genetic rearrangement differs between tumors, so it requires personalized databases. And hopefully from this we can identify some biomarkers and drug targets. And it's likely that as sequencing continues to grow, proteogenomics will also continue to grow, particularly in identifying genomic abnormalities in disease. Does anybody have any questions about this? I think it's all very exciting, personally. But
So, let's say that, instead of I'm not talking about the
annotation.
annotation,
Yeah.
but if we get proteomic data and transcriptomics data, can we also somehow understand the post-translation of what I mean, the regulation after the mRNA levels, like doesn't change the mRNA level, but it changes the
Yes, totally. And that's another thing that I did not bring up, but it's a great point. Yeah, so finding, let's say you see there's no mRNA and there is protein, right? That just means that maybe you have a really stable protein that is not being constantly transcribed. Or in the opposite direction, you have a lot of mRNA and then you have very little protein, so there's some sort of degradation occurring, right? And I think that that's a really important point that you can find from this.
That's done, I mean, this kind of
Yeah, people are definitely doing that. I think it's a little bit new still, but it's definitely happening. Yeah.
So it's in Jessica's example, it's a histone modified is one example of this post-translation.
So I mean that would be that's a sort of another example, right? If you have the histone modification thing doesn't really apply here, but let's say you know that there's some sort of modification on your peptide, and that may be causing either increased degradation or decreased degradation, and then you can use modifications as well as another layer. We also you can use phosphorylation as another layer in this, but that's a whole nother lecture. Yeah. Great.
But I mean, you have to have some previous knowledge about how this. I mean, because I'm still talking about the modification thing.
Right. Go ahead. Yeah.
I'm just curious. Is there any way to systematically recognize this kind of post-translation modification without any previous knowledge?
Yeah, so that's sort of a different thing, but yeah. So what you normally do to find post-translational modifications, and I think that you probably have a lecture on this, a whole lecture on this, but you can in the search tool like Xtandem, that's an option where you put that in. So you don't have to know that that exists, but you have to know that it could exist, and then in the search it adds those modifications in as possibilities. But that's that's part of the search tool that does that. Yeah. How do I'm taking my mic off because I don't know how to do this. Where did it go? Screen flow. Let's see. And shift